What you see here is a scheme to explain principally the neo-Darwinian evolution. That means after the fusion of Darwin's idea of evolution of living organisms together with the genetics, starting with Gregor Mendel. That happened around 1940. On the left side, you see genetic variation. Darwin spoke, of course, not on genetic variation, but on phenotypic variation. That's what one sees or can nowadays measure. According to the theory of genetics, starting with Mendel, there must be some genes behind. And of course, as we know now, the genes were only discovered in the 1940s. I will come to that in a little while. Now, variations are changes in these genes, and my topic will deal with particularly what are the mechanisms by which nature manages to occasionally make some genetic variations. This is the driving force. You can imagine if there wouldn't be any, if the genetic information of living beings would be absolutely stable, there would not be any evolution. But the direction the evolution takes is being taken by natural selection. That means the way how living beings deal with their encountered environment, their habitats. And the habitats are composed of the physical-chemical composition, so non-living conditions, and all the other biological organisms which are in a particular ecosystem in which the considered living being lives. There is isolation, which is a modulator of the evolution, because in geographic isolation, not all the living beings can be found in ecosystem because they may be geographically too isolated to occur there. And then there is the reproductive isolation, which is important for eukaryotic organisms, as we know, which need to reproduce by sexual processes. Now, the question is, how can we study genetic variation and see what kind of changes in the genome has occurred? This is nowadays possible by sequence comparisons with the help of bioinformatic tools. You can look at a whole genome or part of a genome, group of genes, of a single gene, or even of a functional domain within a gene, comparing of different organisms having more or less distance from their evolutionary separation. What one can also consider individual processes to look at when a new mutation occurs, what has been changed immediately, and this is of course the simplest to do that with very simple living beings like bacteria or viruses. And my talk will consider largely on microbial genetics, using bacteria and bacterial viruses. So these are case-by-case studies, and I can already inform you that one sees in doing these investigations that there are more than one specific process at work, and I will describe a few of these specific processes. Now, knowing specific processes, then one can go to the sequence comparison and just hypothesize how it came about to have these different changes. I show you here what we know from bacterial genomes. E. coli, with which much of the genetic studies have been done, has a genome of roughly a little bit less than 5 million base pairs. Later in my talk, I will compare the linear. Here, this, in fact, all these base pairs are just carried in one single chromosome, which is circular. And you see there the base pairs are ATGC, and you see there are genes and there are expression control signals, and then there are, of course, inter-gene regions which also may or may not have particular functions. I define here a mutation as an alteration in the nucleotide sequence. So, for example, changing a G into an A would be a very local mutation. Bacterial cells are, as we all know, are haploid. That has an advantage, that in fact, when cells divide, one cell dividing in two cells under good growth conditions, E. coli bacteria and some other bacteria similarly have a generation time of only half an hour. So within one day you have a very wide population. Occasionally, one of the cells shows to have undergone a particular spontaneous mutation. And this occurs not as shown here already at the time when you have 4 cells. It occurs, the first mutation you can see when there are between 200 and 300 cells. So you see that the rate of spontaneous mutagenesis are rather low. And that must be so, we come back to that later. The principle of life is that the genetic information is relatively stable. Sometimes a mutation can be lethal, there is no progeny. Others are dividing and stay at least for some time in the population. The mixed population, as you see them here, of course, the parental as well as the mutant types are steadily submitted to natural selection, as I defined it just before. Maybe I should just remind you, on the left side in classical genetics, one interpreted a mutation as an altered phenotype, which is for the second condition, is transmitted into the progeny. This definition will have to be careful now, because of what wasn't known at that time. There are some epigenetic impacts which can sometimes also be inherited into the progeny for just one or a few generations. So, but anyhow, it's better not to stay with the left definition. The right one, the molecular one, says that an altered nucleotide sequence. I don't know any mutation, except of course these epigenetic changes, which are not altered nucleotide sequences. If you go with this definition, alteration of sequence of nucleotides, you will see, and there is a wide common agreement among scientists, that beneficial, favorable mutations are rather rare, occurring at relatively low rates and providing a selective advantage. Much more mutations are in fact unfavorable, give selective disadvantage. As I mentioned before, they can be just legal, the cell will not have progeny any longer, or the cell can have progeny but it has difficulty to grow, and that would mean that at long range this unfavorable mutation will disappear from growing populations. What is also important that quite a number of alterations are without immediate influence on life processes. Maybe at some later time, together with still other mutations, they may have some influence, but not at that time. These are silent, sometimes also called neutral mutations. From what I told you, that favorable mutations are quite rare, there is no good evidence for the directedness of spontaneous mutations. That means that a bacterial cell would have a sensor when it comes to a new living conditions, another growth medium, to say, well, that particular gene should be changed in that particular way. That's not how nature proceeds. All these mutations are, to some degree, random and not clearly reflected. It's natural selection which will decide if a particular change is favorable or unfavorable or neutral. It is clear, as I mentioned before, mutations cannot occur frequently. There are various ways to keep these frequencies low. I introduced in that projection a summary of what comes out mainly from microbial studies, but we can see that higher organisms just use the same ways, mechanisms and strategies. I should say that if you study with E. coli bacteria particular mechanisms, it's not said that all other living use exactly the same mechanism. It's remarkably, nature is remarkably inventive. If you look for other living beings, they use in order to approach the same goal, they use often different specific mechanisms, and often in these mechanisms are particular enzymes involved, as we will see. So you see on the left side, again, the sources of mutation. On the right side, natural selection, which I discussed before. By the way, at the very end of my talk, I will also come to the size of our planet. The biosphere is limited to some degree. We cannot have more living beings than there are places on our planet. It has its dimensions. The isolation is also there. So let's concentrate on the left part there. I will, in the following few projections, give you some examples for mutagenization by particular replication infidelity, as I call it. Then there are, of course, chemical and physical mutagens. Some of the chemical mutagens are already internal in the living being, some others are environmental. Then there are a number of recombinational reshuffling processes driven largely by enzymes, and there is horizontal gene transfer, which also needs finally, at least in some cases, the recombination to fix transfer DNA into the genome of the infected cell. Knowing this, and I think you will understand when I give you a few particular examples, we can define 3 natural strategies for spontaneous genetic variation. One is a local sequence change. I give you an example just in a moment. Others are DNA rearrangements of segments within the genome. And the third is to acquire from another living being a segment of DNA which is incorporated in the genome of the recipient cell. So let's see base pairing. In 1953, Watson Crick showed the double helical model by nucleotide base pairing, and as we all know now, that's common knowledge, there are adenine pairs normally with thymine and guanine with cytosine. Now, in the summer of 1953, at the Cold Spring Harbor Symposium, which gave a volume of the presentation, Watson and Crick say that they are aware that, for example, up there on the left side, the standard form of adenine, which is shown there, can change temporarily for a short period of time into an imino form. And you see it's just that one of the hydrogens changes its position within that organic molecule. And that imino form, which is a tautomeric form and short-living, can be paired if by chance the replication fork just moves through at that moment, incorporate not any longer thymine, but cytosine. It fits, it gives a relatively stable base pair. But as soon as the imino form shifts back to the standard form, of course, cytosine is there on the wrong place, it's a mispairing. In the textbooks, you still nowadays read that these are errors of replication. I think we should be aware that nature can use isomeric forms of biological molecules as for activities to be exerted. And this is a good example. It's not never the replication fork incorporated correctly something which base pairs. The problem is that's not the standard most stable form of adenine. And nature, living being couldn't live with that if not repair enzyme systems would have been discovered in practically all living beings in which they have been looked for so far. And they can go just behind the replication fork, see if there are mispairing there, put it back, and this can even identify which is the non-mutated parental form and which is the newly synthesized wrong form of the nucleotide. This is based on maturation within the strand of double-stranded DNA in both strands, and the methylation comes in only post-replication. So after replication, at some time, the enzyme which is able to methylate particular positions of the nucleotides will become. And at that moment, of course, the repair cannot decide which is the parental strand and which the wrong strand, and then the chance of leaving an error behind is 50%. go further then to a list of DNA rearrangements, all done by, mostly done by enzymatic processes, general recombination, which is well known also from classical genetics, which recombines during meiosis maternal and paternal chromosomes sometimes. Then there is transposition of mobile genetic elements. These are small segments which encode for transposase, an enzyme which is able to sometimes under duplication make a novel copy, integrating this copy somewhere else in the genome. E. coli, for example, has a number of these so-called insertion sequences, IS elements, which are transposable genetic elements. And from each of these, there are one to just several of these in the genome. By the way, if you carry these IS elements have a length of about a gene or a little bit more, that means in the average 1,000 base pairs. These are homologies which also provide a substrate for homologous recombination. So homologous recombination occurring between these IS recombinations can give inversions or duplications and transpositions. Similarly, the site-specific recombination are found and have quite particular possibilities, as I will show you in an example in a while, for either gene fusions or operon fusions. Gene fusions are you make a recombination within two different reading frames, and the hybrid, once in a while, not frequently, but occasionally, can have an absolutely new function with having different functional domains combined differently. While the operon fusion is fusing a control signal for gene expression with an intact reading frame and changing thereby the availability of that product, either making more of that product or less of that product. I like to leave the rest not incomplete, therefore there are probably other, we call it, as we don't know them yet, illegitimate recombinations occurring. For the others, the mechanisms are in part at least well understood. You see here an experiment of which I'm quite proud. Think about whether you can study lesion mutations. A lesion mutation makes that the cell will die and not continue to grow. So in order to make biochemical any studies, even of DNA sequence, you need a certain number of copies of these cells. Now, what you see here is a Lysogenic Bacterium. For those of you who are not so familiar with microbial genetics, I just tell you that some bacterial viruses, upon infection of a host cell, sometimes produce progeny viruses, and sometimes the reaction is such that the cell survives, but you can find the genome of that virus in the cell, and these are called lysogenic. In fact, because it is possible spontaneously that the cell growing, the lysogenic cell, all of a sudden becomes induced and produces viruses, gets killed by that and liberates 100 or 200 viral particles. This bacteriophage P1 is maintained in the lysogenic state as a plasmid, a self-replicating plasmid. It replicates in the same, each time the genome divides itself, each progeny receives one plasmid. So it's like the second chromosome in that cell. Of course, much smaller than the host chromosome. Other viruses can integrate into the large bacterial genome like an IS element. These black things are symbolically IS elements. Now, if you grow this T1 lysogen for a long time, I will show you this experiment, what we have done for a quarter of a year. Every morning, except on Sunday when we were not in the lab, we divided heavily a growing culture, and the next day it was thrown up, divide again, dilute again, and let it grow up. So after that long period of growth, some of these viruses couldn't be induced any longer, neither spontaneously nor by tricks like ultraviolet radiation. So apparently, so there was no production of viruses, but we could show that the plasmid was still there. So if you then grow the cell up and isolate the plasmid, you can see. And interestingly enough, in 95% of these spontaneously occurring illegal mutations, an IS element jumped into an important gene. There are about 50 genes to produce these viral particles. If one of these genes was killed, then you don't get viral particle progenies. Only 5% were local mutations, substitutions or other very small local. So that shows that these IS elements are really often, of course, also killing the bacterial cell if they transfer. You don't see so easily if one cell is lost in 1,000 cells, that doesn't matter so much for a bacterial culture. So, having a certain number of these isolates, we could map them in the genome of T1, which is shown, this 90,000 base pair genome of T1 is shown linearly, and each dot shows in which region such IS elements were found. In each point there is an independent isolate. And interestingly, you'll see that it's not randomly distributed. There are hot regions, and there are regions in which, in this experiment, we have no integration. Most often, IS2 was found, less frequently IS30, IS1, IS5, and gamma delta. These are the blue ones. This area there, which you see where so many IS2 and all the IS30 were integrated, were then sequenced very precisely, and for all the sequenced IS2 insertions, none of them was found exactly on the same place. In contrast, the 3 IS30 were all found between the same 2 nucleotides, and 2 in one direction, 1 in the other direction. I said to you before, nature is very inventive. There are a lot of things which nature does. You believe all of these function the same way. That's in no way like that. And we have still no idea what attracts so many IS2 in this particular area there, in which each one is at a different site. So that's not site-specific, it's kind of regional specificity. There must be some regional characteristic in that sequence which we couldn't identify, which attracts so many IS2 in that area, although there are others elsewhere also. Yes. I would like to give you just a short introduction into site specific DNA inversion. What you see there is a 26 base pair long sequence which has right in the middle are 2 A's, so that gives it some direction. Part of this flanking areas are homologous in different orientation. As you see, some others can be any nucleotide. N means it can be anyone. That's what we call not a specific sequence, but a consensus sequence. The enzyme looks for these particular sequences, and if 2 of these sequences are carried in inverse orientation, as is shown over there, with a particular reading frame and directional promoter for gene expression, you can see that inversing the thing, then the reading frame gets changed, but it has no promoter any longer. So this is a flip-flop on-off, which is found for proteins in the bacterial surface sometimes. It can be found in viruses also. Similarly, on the right side, one of these consensus sequences is carried here within a reading frame for, in fact, a protein which determines from that P1 virus which we have seen before, the host range. The host range is depending on the end of the so-called phage tail with tail fibers looking for appropriate infective hosts. So the host range of VC in this combination is different from the host range shown down there by V prime C. And that, if that goes back and forth, you'll get each time a different population of host range progeny if the tail fibers are like that. So you see, this is an example for gene fusion, fusing different functional domains, while the other is an expression for bringing a particular gene under different expression control signals. We have asked at that time whether if you would have in a DNA molecule, this is a relatively small plasmid which was constructed particularly for that example by Shigeru Ida. And the consensus here is a little bit different from what you have seen. In color and large letters, the classical consensus. These others are in this particular experiment. We have it only once there as Jigs star. When you the plasmid was such that there is a kanamycin-resistant reading frame that would give resistance to the antibiotic kanamycin, but it has no expression control signal in front. This P blah is ending, this transcript is ending rather rapidly by a functional terminator. So it doesn't read through into the kanamycin resistant gene. And if you grow for a long, long time, you find perhaps 1 in 10 to the minus 5, 1 into 10 to the 5 cells which have become kanamycin resistant. And if you look in this experiment, we could see that there was DNA inversion between Jig star and any of these letters A, B to K, and these were finally sequenced. What you see is that if you take these sequences from A to K, there is nothing absolutely necessary which we could identify for being a so-called secondary crossover site, which serves very occasionally only for DNA inversion. But it gives rise, of course, this mechanism gives rise to create fusions, and some of these fusions may be functionally really new things and improvements. You see that in this experiment, what you see here, these numbers give the number of independently isolated secondary crossover sites which were found independently. So many are found once, a few twice, one 5 times, and one 6 times. There is some statistical reproducibility, but any of these secondary crossover sites serve only at very low frequencies, while the consensus, the good consensus, of course, is much more efficient. Every few generations you have that inversion. So my idea is that probably the flip is often occurring flip-flop, as if it's none of the two regions are in fact bad and giving disharmony or lethality, then a flip-flop is tolerated, and that these enzymes are probably more important for making these rearrangements within the genome at very low rates for creating novel things for the biological evolution. I will come to that later on, kind of with the claim that nature has learned to evolve in living being, and this is the source of our very much appreciated biodiversity. The last of the 3 natural strategies is horizontal transfer. Transformation, conjugation, and virus-mediated transduction were discovered and described for the first time in the 1940s. Transformation was a proof that with well-purified DNA molecules from a donor mixed with recipient bacteria, that they could penetrate and change some phenotype. So that was then a clear proof that the genetic information is carried by double-stranded DNA molecules. The double-strandedness and the structure altogether was only shown by Watson Crick, as I mentioned before, in the early 1950s. So about 9 years after these transformation experiments carried out in Rockefeller by Avery and his colleagues. Conjugation needs cell-cell contact between donor and recipient bacteria, and in virus-mediated transduction, the viruses transfer a segment of DNA. There are various ways, again, nature being inventive, that can be done. Some of the viruses, like P1, package into an envelope of the progeny virus, which is still empty. DNA usually just to fill ahead one of these envelopes fill, and then it's finished. That's just the size of the genome of that P1. But sometimes they start to package on the bacterial genome. So this viral particle is full of just bacterial DNA of relatively small size, of course. Other viruses, like bacteriophage lambda, who in the Lysogenic Bacterium is integrated as a part of the genome of the host cell, then sometimes the excision of that genome, when phage starts spontaneously to reproduce, then sometimes the crossing over is not done very precisely at the end of that viral genome, but elsewhere. and that can give viruses which are hybrids, having a part of viral DNA and part of host DNA, and upon infection gives that DNA to the infected host. Of course, these are usually defective because some viral particles are left behind, but it gives the idea to use plasmids on the one hand and viruses, viral DNA, as vector molecules, which gave later on rise to the strategies of genetic engineering. I will come at the very end, very shortly, to the genetic engineering aspects. There are a number of factors limiting the acquisition of foreign DNA. The surface compatibility must be appropriate. DNA must be able to penetrate through the cell wall. Once it can encounter restriction modification systems. And finally, if everything functions, the gene is propagated and expresses its functions. That sometimes destroys the harmony of functional harmony of the cell, so there is no survival. But often the functional harmony is not disturbed, and in fact can give it a new property. We have learned a lot for these horizontal transfers by studying antibiotic resistant genes. So I mentioned that earlier this afternoon, this morning. I was just, some of you may have had a chance or your future time to work with restriction enzymes. When in the around 1970, the first enzymes were isolated, it was becoming pretty soon clear that not all systems found in bacteria work in the same way. EcoR1, for example, which is shown there on the top, is the so-called type 2 enzyme, which reproducibly cuts within the sequence which is recognized. In this 6 base pair sequence, you see 2 adenines, one on each of the strands with a star. That means in the cell which carries the restriction enzyme, these adenines carry each in each strand a metal group. The metal group is brought in by methylating enzymes, as I mentioned before, shortly after the replication. And when the restriction enzyme type 2 encounters such a sequence, GAA-TTC, fully methylated or only methylated in one of the strands, then it is protected against restriction. The restriction enzyme cannot cut. But if it's foreign DNA, it may well have, usually they have metal groups elsewhere, but not in this particular sequence. The restriction enzyme is discovering that, cuts into fragments. So fragments are made by all penetrating DNA not appropriately labeled in that particular sequence, by methylation, and then the fragments are relatively rapidly chewed up by exonucleases. These are other enzymes starting at 3 ends of DNA and putting them back into single nucleotides. But during that short period of time, when there are a fragment with open ends, they are recombinogenic. They try to find a way to incorporate into the genome, and occasionally one of the segments is finally found in the host genome. So that's for horizontal transfer. The type 1, which we had originally studied, are different again, but the function is similar on the very end. You see here in equal R1, equal K1, the sequence is not 6 but 7 nucleotides. These are on the colors. They are separated by, it must be 6, not 5, not 4, any nucleotide, just spacing these 3 from the 4. And again, an adenine in each strand becomes methylated in the cell's own DNA that protects the cell's own DNA against these restriction enzymes. And interestingly, once an enzyme identifies that sequence, it gets activated and it rots through the DNA from both sides, still fixing the protein there, and one sees these growing loops of DNA in the electron microscope, and so I've drawn that as migration. It's not, the enzyme doesn't migrate along the DNA, but it stays there but rots the DNA through. If there are more than one side, the roping goes on both sides, not only I've shown here on one side. Then all of a sudden the two migrating complexes run into each other, stop, and then it's cut. And the fixing of an enzyme down here or up there is not synchronous, of course. Therefore, each time the cutting site is at a different time. For experimental studies using restriction enzymes, of course, only the type 2 are important because it's reproducible. You cannot use these very similarly so these are not on the market. But nature uses various ways to do it, and for evolutionary purpose, this is in fact better, because there, if that small 6 base pair recognition site is each time cut, it may be inside an important gene which the cell never can acquire because cutting is rather fast. While here, all the genes have a good chance temporarily to survive until the chance of being incorporated in the genome is there. So it's interesting. There are more types which I haven't shown. Type 3, which stretch out an arm and so on. Good. A few conclusions now from what I told you. Charles Darwin said already that living beings come in a long period of past evolution from common origin. He hasn't said one or how many origins of life there are. We don't know. In fact, all what you tell you here is how evolution nowadays occurs in all living beings which we have studied on this planet. We still have only hypothesis how the first living being came to life, but there is no experimental proof how the start really of life on planet Earth has occurred long time back. We can, however, now project that we can say Darwin was right. The living being nowadays among us have common origins. We can extend that conclusion. The future life of all of these have also common future, because occasionally any of these growing branches will have a chance to receive one or very few genetic important functions developed elsewhere in that tree of evolution. That's wonderful, I think, that we exchange occasionally, and natural selection decides whether this is appropriate. If it's helpful, it's maintained. If not, just forget about it. Another important thing, I defined here the local strategy, the DNA rearrangement strategy, and the DNA acquisition strategy. I explained you by examples of some of them how it functions, and on the second part of that projection, you see that the local one is giving sometimes, not reflectively, sometimes an improvement of available functions just by changing, for example, one amino acid by another one in the gene product or so on. Also by changing some efficiency to positive or negative in expression control signals and so on. There are also in the local sequence changes possibilities that sometimes one nucleotide or a few adjacent nucleotides are all of a sudden missing, or one is additionally inserted, or a few others are inserted, or the nucleotides are a little bit intermingled. These are all local changes, and this is still valid to give a molecular clock to see how long back two different organisms which are compared have separated from each other on the evolutionary tree. Although we have to be aware that the other strategies are not included in the molecular clock. And I showed you that IS element transposition is an important source of leading mutagenesis, for example. Now, DNA rearrangement gives an improvement of available now capacities, as I mentioned, by gene fusion or operon fusion, still other processes. And the DNA acquisition strategy is a sharing in successful development made by others. And that's the most efficient acquisition of new activities. If they help you, then it's fine. In the presence of antibiotics in a patient or in the environment, of course, it's good to acquire horizontally a plasmid or by some other way antibiotic-resistant genes, and that occurs once in a while. And it is, of course, still a big medical problem, as it has been discussed earlier today. I guess that these two strategies here, the intragenomic rearrangement and the DNA acquisition, can explain some of these emergences of novel properties, of which many evolutionary biologists still have no clear idea how that could occur. But this is a good source for these things. This is another philosophical conclusion. There are evolution genes, which are genes for rearrangement, for restriction enzymes keeping the rates of horizontal transfer at very low levels. I mentioned before the repair systems. In fact, they are not needed for bacterial growth from one generation to the next. They serve evolution, and I started to call these evolution genes. Their products are really very essential for the biological evolution. There are, but however, not only products of evolution genes, non-genetic elements are also implied. These can be structural flexibilities of biologically active molecules. I showed you the automeric forms of nucleotides, which contribute to evolution. Nature uses these capacities of organic chemistry really to give appropriate modifications. And by keeping the rates low by various means, as I mentioned, it is tolerable. random encounter. For example, if a virus infects bacterial cells, that's a random encounter, and it may carry by chance a segment of the former host chromosome. Environmental mutagens also, of course, are there, and nature uses all of that. And that means taking these things together, that the reality found in nature takes actively care of biological evolution. And this is an expansion of the Darwinian theory to the level of molecular processes. Very briefly, two antagonistic principles. On the one hand, we have genetic variation, which helps to accelerate and undergo biological evolution. On the other hand, limitation of genetic variation to provide genetic stability, preserving particular species. We believe this is not so easy to show, that all these evolution genes have been fine-tuned for their job in the long past of all living beings in the evolutionary development since life exists on our planet. Here, that's another philosophical interpretation. The genome has actually a duality, and the genome of bacteria has actually a duality, of all plants, of all animals, of us human beings. On the one hand, of course, many genes help the individual to come from one generation to the next. These are housekeeping genes, accessory genes of use under particular life conditions, that you can live under different life conditions. And for the eukaryotic organisms, of course, developmental genes, which are important for the individual lives. Then there are a smaller number of genes which principally are there for evolution as generators of occasional DNA sequence variations and also acquisition of genes. But particularly in higher organisms, some of these products serve both purposes. When they are there, they can serve. For example, mutagenesis can occur not only in the germline, where it's more favorable, actually it's more often in the germline, than in somatic cells. It can, in somatic cells, for example, be the cause of cancer. So we are aware of that, but nature tries to keep all that at a very low level. It's not always so successful. I think if it would be too strict, there wouldn't be any evolution. You know, if we keep it to zero, then there wouldn't be any life development possible. The individuals are for the fulfillment of that particular life, while the evolution genes are here for the expansion of life, different types of life, biodiversity. And we suffer from losing biodiversity. It is clear that thanks to these powers, potency of nature to undergo evolution, biodiversity will always be regenerated. Not the same as we have lost. We have to be aware. There are so many possibilities. So that's just don't believe that if we lose a lot of biodiversity within 100 years, that within the same 100 years we will get the same level back. That's a completely different time scale, a very long time scale. So one has to be really careful. To finish, I like to mention that I have been motivated in the early 1970s to study molecular Darwinism because of the upcoming genetic engineering thing. In the Asilomar conference held in February 1975, this was a conference held by scientists to discuss potential risks of genetic engineering. And it was concluded there that there were, of course, if you work with genes, gene products, in the laboratory you can infect yourself. As it was known since long time that microbial laboratories for medical microbiology have developed very protective working rules for not infecting themselves by the pathogens which they study. It is possible. So it was said that we should, any gene which you isolate and start to manipulate, study, you should take it as potential potential for toxicity or pathogenicity. So apply careful methods in the laboratory. Try not that it doesn't evade from the laboratory. But at that moment, it was already said that if at some later time recombinant DNA would be by purpose or by accident liberated into the environment, the question is, what would these manipulated genes, so-called manipulated genes, spread into other living beings? And that made clear to us that we need to understand Darwinism at the molecular level. In my lecture, you have seen that we have learned a lot. Conclusions were that gives very good evidence for the importance of evolution in nature and how that works, usually in small steps. And what you see here is that genetic engineering is just applying the three same strategies. You can take from a donor cell a segment of DNA which is carrying one or a few genes. This may be perhaps up to 1 page of a book. In the genomes of bacteria, I showed you that E. coli has about 5 million base pairs. That's a rather important book, in fact, corresponding roughly to the Bible, Old and New Testament together. Some other bacteria have a bit smaller genomes, but still corresponding to a book. Higher organisms, yeast has already 3 books of this size of E. coli, and higher organisms, often several hundred, up to 1,000. Human beings, estimately, in this comparison, have 700 volumes of an encyclopedia in their genome. So what you can do is transfer a page or a fraction of a page into that of something which you know what it does, and that's exactly how horizontal gene transfer works. If you would try to combine half the genome of an apple tree with half the genome of a strawberry plant, you would certainly not get a tree having strawberries instead of apples. That doesn't function. The harmony of that hybrid would be absolutely zero. Now, you can also extract some DNA, make site-specific mutagenesis. In order, that's often done to inactivate a particular gene to see what the phenotype is if you activate. Then you learn the functions of these genes, you introduce it, and so on. So, and also you can, rather than local mutagenesis, rearrange or duplicate or delete a segment of genome. That's what is usually done. So there is no principal difference between what nature does since always, with long periods of time, and that is also done by classical breeding techniques in plants and animals. And we do know from natural processes and breeding techniques that chances of unexpected accidents, dangers, and so on, unpredictable dangers, are not existing or only to a very minor. And we can expect that genetic engineering has the same level of chance of unexpected accident, of horizontal transfer, and so on. Now, with this knowledge, biotechnology has already, and is still proceeding from a classical way in which if you want to take advantage of biological functions, classically you went into the nature, you looked for an organism and used that. Agriculture has its origins in this, just looking which plants can serve for our food and so on, and similarly with animals. And then more recently, when it became clear that, for example, radiation causes mutation, people started to strongly irradiate, for example, wheat in the fields by hoping that there will be mutants which give higher yields and so on, just selecting for those few grains which gave higher yields. But it was not possible for a long time to sequence to see what other mutations are there which you didn't see any phenotypic manifestation. While in genetic engineering, you usually, if you are careful enough, you have your plan, you do the experiment, you look at the very end whether the new manipulated genome is really what you expected. Genome sequences is very routine nowadays, and that should in fact be done. And what one can do in modern biotechnology, explore the biological functions that may serve your purpose. You may, by genetic engineering, improve by site-directed mutagenesis the reading frame if needed, or having a better expression control signals to get higher yields. And you can introduce the whole segment then of DNA into the most appropriate host. You have to be aware that if you do that, finally you have some combinations which are not viable, so those, of course, you can forget about, but the others are like that. It is, since some time, of course, possible to produce, coming back to a former presentation, human insulin, not only taking that from human beings directly as it has to be done for long periods of time, but you can transfer the appropriate genes into bacteria and produce human insulin by biotechnological means. So this is just one very positive example, and this will keep going. To finish, I'd like to show you a projection and shortly discuss it, which I have shown a few years ago in China at an international meeting of biotechnology. This is a roadmap for agricultural biotech. As we all know that our food is composed on animal cells, but largely also on plant cells. And it is possible nowadays, with all our increased knowledge on metabolomics, to see how our metabolism functions, what are the factors which we need, and exploring whether the food plants which are at our disposition have these possibilities. Some do not. And golden rice, which was developed some years ago and will be becoming planted pretty soon, I guess, is a good example that it is possible to provide to our daily food also precursors of vitamin A, and these precursors we can transfer them into vitamin A in our body, and so that is really, to my mind, a very important development which is coming up and which would help also in medicine, because worldwide about 15 or so% of the world population suffers from malnutrition, undernourishment, and so on. So if we succeed to bring in a certain number of genetic information for all we need into just, let's say, a handful or 2 handfuls of food plants, being our daily diet, I think the medicine will have a very good future. Not from 1 year to the next, but I would say probably 1 to 2 human generations. That's my view of where medical improvements would go. Thank you for your attention.